Management of waste cooking oils is a global environmental and urban infrastructure problem. It has been reported that the improper and uncontrollable disposal of this waste in domestic or industrial pipes generates extraordinary levels of contamination in water bodies that damages the environment and causes diseases in vulnerable rural communities. Despite the significant global relevance of this problem, it continues to be underestimated in developing economies such as Mexico's, since, despite various efforts by its Ministry of the Environment to promote sustainable management of this waste, the country still lacks standardized recycling systems. Currently, in the biotechnology sector, waste cooking oils have had great relevance as a potential substrate for the production of biodiesel, biosurfactants, biolubricants, and, as in the case of our project, biopolymers such as PHA, polyhydroxyalkanoate. PHAs are a group of polymers naturally produced by microorganisms with an attractive economic importance in various industries due to their syntactic, hydrophobic, and piezoelectric properties. PHB, poly-3-hydroxybutyrate, is one of the most widely used PHA polymers in various industries, such as biomedicine, sustainable packaging, and agriculture. However, the current industrial cost of PHB using native producing bacteria is 5 to 6 times higher than that of petrochemical plastics. Therefore, different approaches have been proposed to improve its production. Among non-native producing organisms, Escherichia coli has been commonly used on an industrial scale. The production of PHAs in recombinant organisms shows strong limitations, including cell volume, modulation of molecular weight of polymers, and purification. Wu produced PHB in a recombinant E. coli that generates elongated cells that can hold a greater amount of PHA polymers. However, accurately controlling molecular weight, which determines mechanical properties and degradation rates, remains a challenge. Polyethylene glycol, PEG, has been identified as an effective chain transfer agent for modulating PHB chain length. In addition, coenzyme A, CoA, has been considered as an inherent inhibitory mechanism of PHB elongation. However, a comprehensive kinetic framework for PHA elongation remains absent. No mathematical models quantify PHB molecular weight dependence on PEG/CoA concentrations or define the theoretical maximum chain length under specific biological conditions. Finally, PHA purification is extremely laborious, so we will use autolytic bacterial systems which have been widely proposed as an alternative to extraction with chemical solvents, such as chloroform or dichloromethane, or mechanical breakage, such as high-pressure homogenization or pearl milling. With this in mind, our team identified several areas of opportunity in our project. First, utilizing waste oils from the northeastern region of Mexico, where there are currently no fully established systems for their treatment and use in circular bioeconomies. Second, proposing a novel system to modulate polymer length in recombinant systems. And third, using an autophagic system as an alternative for the capture and purification of PHAs. With this project, we hope to boost the bioeconomy of our region based on the feedback from our principal investigator and advisors who helped us from the project ideation to the formulation of the final product.
We also want to mention and acknowledge the people who supported and guided us through this project. First, Dr. Jose Claudio Moreno Rocha, our primary advisor and main mentor. He guided us through the synthetic biology side from start to finish, helping us shape the approach, refine ideas, and stay aligned with solid scientific direction. Second, Mr. Mauricio Reyes Elizondo, who was a key mentor by far on the computational, synthetic biology, and modeling side. He supported us with the mathematical framework, genetic circuit ideation, help us understand the concept behind the equations, and provided guidance across implementation details, including code development and technical visuals. Following with Dr. Maria Elizabeth Aleman Huerta, a microbiology specialist who gave us valuable practical context. She welcomed us into her lab where we explore microbiology techniques related to PHB production, which help connect the modeling work to real experimental reality. And finally, Dr. Abraham Ruiz Robles, who supported us through the project with guidance and feedback, helping to strengthen our overall understanding and direction.
In August 2025, the original idea we had for the project was evaluated, which was to produce PHA from Nehayote, a byproduct from the nixtamalization process. This because, as we were researching, we came across a scientific paper led by Dr. Maria Elizabeth Aleman Huerta from Faculty of Biological Sciences, Institute of Biotechnology, head of Laboratory 3, focused on producing PHB from organic residues, most notably the chayote. We spoke to her, and she told us about the limitations with the use of it, since the substrate is too complex due to its variability between different types of carbohydrates, such as varieties of cellulose, starch, glucose, a certain percentage of lipids, and some minerals such as magnesium. So we looked for information on methods to purify it. By the end of September 2025, we had our first face-to-face meeting with all the members of the team, in which important dates and points of progress for the project were established. By this point, we had already information about PHA and its production in bacteria, but we still had doubts about the best option to use in terms of which organism, substrate, and metabolic pathway to use for a proposal, so the meeting was very useful. And, based on the information collected, as a first decision, it was decided that our chassis will be Escherichia coli. From there, we dedicated ourselves to investigating metabolic pathways for the production of the biopolymer. And it was during that search that we found the article that will be a key piece in our proposal, since they proposed the modification of the cell division of E. coli, so that, instead of dividing into two, it divides into multiple cells, which could help us to generate much more PHA, regardless of the pathway to be used. Consequently, we started looking for a substrate that would serve us as a source of carbon for the bacterium, and we ended up choosing fatty acids as a substrate. But because E. coli does not produce the polymer naturally, we will focus on manipulating its beta oxidation pathway to produce the polymer from acetyl-CoA, taking into account that the best option for the manufacture of sutures is the long-chain PHA, and that there is a direct relation between the fatty acid to be used and set length. At the beginning of October, we had our second face-to-face meeting, in which, together with all the data collected in terms of substrate, organism, and metabolic pathway, we unified the proposal. However, Dr. Alman told us about an important problem in terms of PHA recovery, because these intracellular granules cannot be easily secreted due to their large size. Then, Dr. Claudio proposed the use of autolytic bacterial systems to solve this problem, which is the controlled expression of lytic genes with the aim of disintegrating the cell wall and thus releasing the PHA. Starting in mid-November and throughout the holiday period, we began virtual meetings on specific objectives, which were divided into genetic circuits, metabolic modeling, molecular docking, and kill switch circuits, in which we managed to obtain essential point values, such as the minimum molecular weight in a PHP chain to become a medical suture, the metabolic model that we will be using for the production of PHB in E. coli, and many other more. And finally, it was during this period that, based on the guidance of professors from the Faculty of Physical and Mathematical Science, Michelle Abraham Ruiz Robles, as future perspectives, we chose to carry out subsequent analysis of electrospinning simulations with the PHB. Our project objective is to perform a computational simulation aimed at the optimized production of high molecular weight PHB in Escherichia coli under different lipid proportions, emulating the variability present in residual oils. We will use the recombinant organism E. coli, proposed by Wu, as the chassis of the project. The simulation will be carried out through a system of differential equations that integrates genetic expression, enzyme kinetics, and metabolic data to provide an accurate estimate of the length of the PHB chain achieved. This model will allow the identification of the optimal conditions and genetic modifications necessary to maximize the synthesis, laying the foundations for a future experimental implementation. Finally, we will estimate the commercial viability of the PHB obtained through cheminformatics and token simulations to obtain its mechanical and biological properties.
To see if PHB polymers work for medical sutures, we ran a digital screening of several chemical descriptors. We focused on four key metrics: asphericity for fiber alignment, log B for fluid absorption, ester density for degradation, and most importantly, the radius of gyration. This last parameter is the cornerstone of our study. It allowed us to deliver a key structural value in polymer science. In this graph, the slope of the line reveals the scaling exponent. More than just a number, this value serves as the molecular fingerprint of the polymer's flexibility. It tells us mathematically if the chain is collapsed like a sphere or extended like a rod. In the world of sutures, we look for a sweet spot of 0.6. This value represents the ideal balance, a chain flexible enough for a surgeon to handle tissue smoothly, yet possessing the structural integrity to ensure the knot maintaining this stretch. Our results place PHB at the scaling exponent of 0.75. While this confirms the material is functional and structurally sound within the 100 to 240 kilodaltons range, it also indicates a slight stiffness compared to the ideal. After reviewing PHB's potential with Dr. Michel Abraham Ruiz Robles and considering their suggestions, our next step will be developing medical sutures in copolymer conformation. This approach will allow us to tone that slope toward the ideal 0.6, optimizing the material's flexibility to outperform current synthetic standards and establishing PHB as a high-performance polymer base. To ensure our PHB sutures are safe for human use, we conducted a biocompatibility analysis through blind molecular docking. Our primary goal was to predict how the polymer interacts with critical skin and immune system proteins. A disproportionate interaction with the skin proteins could hinder degradation, while high affinity with immune receptors might trigger unwanted inflammation. For this analysis, we digitally constructed PHB and PGA change, our clinical control, using the RDKit library, simulating their polymerization step by step and optimizing their 3D structures with open bubble to reach a minimum energy state. Then, we tested these polymers against 8 protein data bank receptors associated with distract cellular matrix and tissue regeneration. Then, using Autodoc Bina, we simulated thousands of interactions across the entire protein surface at a physiological pH of 7.4. This process allowed us to confirm that PHB exhibits a balanced and safe affinity, validating its potential to be integrated into the body without eliciting adverse immunological responses.
For the modeling part of the project, we built an in silico framework to predict how genetic design choices affect gene expression, cellular stress, and ultimately polymer chain length and yield, which matters a lot for applications like medical sutures. At a high level, our system has 3 key genes. First, PHA-A and PHA-V are the monomer supply side. They help generate the precursor substrate. And PHA-C is the polymerase, is the enzyme that actually links monomers into the PHB polymer chain. So the design goal is not just to get more expression, but we specifically want expression balanced, so the substrate supply is abundant when polymerization happens. Our modeling pipeline follows the biology in order. First, transcription dynamics, how much mRNA we make for each gene. Then, translation constraints, how ribosomes limit protein production and create stress. Then, polymerization kinetics, what controls polymer chain length and how we can predict it. And finally, we apply host constraints, like how much PHB can physically fit inside the cell before it becomes unsafe or inefficient. Now, we model transcription using a deterministic ODE taken from IM FCB-UANL 2021, basically tracking mRNA concentration over time, as production minus degradation. Production is controlled by induction and promoter behavior, and degradation is modeled as first-order decay. This gives us a clean way to compare the signs at the mRNA level, which is the base layer of everything else. Then we tested gene dosage with 2 plasmid backbones, a low copy plasmid at about 25 copies per cell, represented as capital letter L, and a high copy plasmid at about 200 copies per cell, represented as capital letter H. We also separated control using the 2 orthogonal dosable systems, IPTG for PHA-A and B operon, and vanillic acid for PHA-C, so we can independently tune substrate supply versus polymerization. These inducers play a role in the genetic circuit design. We designed 2 inducible plasmid circuits controlled by 2 different inducers, so we can activate them sequentially instead of all at once. The first circuit contains PHA-A and PHA-V under an inducible promoter. This model is responsible for building up the precursor pool, basically generating the substrate that PHA-C will later polymerize into PHB. So operationally, we induce PHA-A and PHA-V first, near the end of the exponential phase, to let the cell accumulate a high concentration of precursors. Then, only after the precursor pool is high, we induce the second circuit, PHAC, which encodes the PHB synthase. This timing matters a lot because if we induce PHAC too early before enough substrate exists, the enzyme starts polymerizing under substrate limited conditions, which tends to produce shorter chains. By separating induction in time, we aim to ensure that when PHAC turns on, it immediately has abundant substrate available, favoring longer chain PHB. Next is translation. Even if we produce lots of mRNA, the cell has a final ribosome pool. We use a ribosomal saturation model with a total ribosome pool on the order of 50,000. The effective translation rate behaves like a saturation curve. It increases with free ribosomes, but it can grow indefinitely. Finally, polymerization. We model PHB formation as a chain growth process, and the key metric is degree of polymerization, or DP, which is basically how many monomers are in an average polymer chain. In our model, DP is controlled by a competition. Propagation grows the chain, while termination and chain transfer cut it short. That trade-off is captured by the equation where 1 over DP increases when termination or transfer increases and decreases when propagation dominates. Here's intuition of the derivation for the polymerization equation used. We describe polymerization in 3 steps: initiation, where enzyme becomes active; propagation, as chain elongates; and termination as change is released. Termination can happen by spontaneous dissociation or by chain transfer to an agent like PEG. The key modeling attribute is that we use the reciprocal because termination competes with propagation. So instead of dP directly, we work with 1 over dP, where each termination pathway becomes an additive term. This makes interpretation and fitting much more easier. When you simplify the expression, the enzyme concentration cancels out, and you get a linear relationship. 1 over dP is equal to a constant times PEG plus a constant. So PEG becomes a direct control knob. If you increase PEG, you increase chain transfers and you reduce dP, meaning shorter polymer chains. But in vivo, PEG isn't the only thing that can cut chains. We also incorporate CoA mediated chain transfer because free CoA can act like a nucleophile and clip the growing chain from PHAC. This is basically thiolysis, and it naturally limits maximum chain length. So the final form adds a metabolic feedback term. Before using this equation in our workflow, we did a quick sanity check using real published data. We took experimental molecular weight values reported with PEG 106 and used them to simulate what the model should predict. When we compared the model versus experimental, the match was very tight. At 0% PEG, the model predicts 268.7 kilodaltons versus 267 kilodaltons experimentally. And at a 1% PEG 106, it predicts 30.4 kilodaltons versus 30.0 kilodaltons, both with around 1% error. Moving on to results, we first simulated 8 genetic configurations to see the ratio between synthase mRNA PHAAC, monomer supply mRNA PHAAA, and V. The important takeaway is that matching everything doesn't help. For example, configuration 8, high copy for both PHAA and V and PHAAC, lands close to a 1-to-1 ratio, which means too much synthase relative to substrate, so you initiate many short chains. For configuration 7, high copy PHAA and V, with low copy PHAAC creates the bottleneck we want. Monomer transcripts exceed synthase transcript by 8x, which favors longer chains. Visually, these plots show the same idea. In configuration 7, monomer supply dominates and synthase is limiting. That's good for high molecular weight polymers. In configuration 8, synthase rises too much relative to supply, so the system tends towards lots of short polymers instead of fewer long ones. Then we check if the cell survives the design. We estimated metabolic burden as a percent of transcriptome capacity required across 3 media conditions: minimal, intermediate, and rich LV, where LV reduces burden the most. Here what stands out. So configuration 8 becomes extreme, up to 92% in minimal media and 79% even in rich media, which is predicted to be lethal or unstable. Configuration 7 is still high, at about 72% in rich media, severe but potentially manageable under controlled conditions. But configuration 1 stays under 32% in rich media, so it's the most stable from a viability standpoint. Next is ribosomal stress. We define a ribosomal stress index, RSI. That's basically the fraction of ribosomal capacity captured by our synthetic system. The pattern is a metabolic volcano effect. So if we crank translation hard for everything, the system collapses. A strong RVS on both PHAAV and PHAC pushes RSI above 80%, which would essentially shut down normal self-growth. A better balance is a strong translation for both PHAA and V to keep substrate supply high, but weak or moderate for PHAC to avoid ribosomal overload, keeping RSI low. Now, the key product property, polymer molecular weight, which we use as a proxy for chain length and suitability for sutures. Based on literature, the target molecular weight window for medical sutures is roughly 100 to 240 kilodaltons. Below the range or above the range, it becomes too rigid or flexible for the intended application. In our predictions, glucose does not reach the target window and stays below the range. In contrast, lipid-based carbon sources, especially residue 1 and 2, derived from regular use household oils scenarios, are the ones that can push the polymer into the 100 to 140 window, meaning they are the viable candidates for producing suture-grade PHB in our models. However, there's a trade-off. Those same lipid residue conditions also tend to drive higher intracellular accumulation, which increases volume occupancy and can prematurely end production or destabilize the cell. That's why the next step is addressing the volume constraint. So even if we get the right molecular weight, the polymer takes space. We impose a safety constraint based on literature that if PHB occupies more than 80% of intracellular volume, the cell is predicted to fail or die. The plots show carbon sources can drive PHB granule volume dangerously close to that limit, meaning production could stop early or even rupture cells. So we applied an optimization strategy. Instead of only changing metabolism, we changed morphology. We modeled a mutant strain that suppresses mutation, creating an elongated giant cell phenotype, increasing cell volume from about 1.1 micrometers cubed to about 5.2 micrometers cubed. That gives more internal storage capacity for PHB without changing the core pathway kinetics. And this is the payoff. In the wild type simulation, production plateaus at around 96 hours because the cell hits the 80% volume limit, capping yield short. But when we implemented the stable low burn configuration inside the mutant strain, the model predicts about a 4.7 full expansion in storage volume and yields at around 128 grams per liter, staying under the safety threshold. This table summarizes predicted yields and productivities across conditions, all while keeping volume utilizations under 80%. To summarize, we built a layered model from transcription to translation to polymerization and used it to evaluate designs that balance polymer chain length, cellular stress, and final yield. A key conclusion is the lipid-derived residues we tested are strong prospects. In the model, they consistently produce long-chain PHP in the range needed for medical sutures, unlike glucose, which falls short. So these residues emerge as the best candidates for reproducible long-chain PHP production, as long as we also manage the physical bottleneck of intercellular storage. That's why our final optimization step, expanding effective intercellular capacity through the non-septating cell strategy, fits naturally into the model, as it helps keep the system productive while still targeting suture-grade polymer properties, utilizing waste oils. Win-win situation all around.
Our main goal was to measure how carbon moves from a complex mix of lipids to the production of important biopolymer building blocks. So, to validate the thermodynamic and stoichiometric feasibility of the system, we developed a genomic scale metabolic modeling framework based on a performance of the flux balance analysis of a recombinant Escherichia coli to evaluate the potential of waste cooking oil as a high efficiency feedstock. Our main goal was to measure how carbon moves from a complex mix of lipids to the production of important. The design of the engineering chassis used two main strategies. Based on the literature, first we made the cells longer by blocking the zeta ring using SUL-A gene overexpression, stopping normal cell division and causing the cell to grow as long filaments, creating larger cells with more space inside. To make use of this extra space, we added the PHA-ABC operon described by another author. This pathway provides the enzyme needed to capture the high carbon flow from beta oxidation and turn it into viable PHP in collision bodies. Before we start the simulation, an important part was setting the biomass growth rate to zero. This step is needed to find the highest possible stoichiometric efficiency of the chassis, setting the biomass flux to zero mimicking the stationary phase. When cell shifts all the resources to the PHA pathway. To carry out the objective, we compared two cases. Residue 1, which was meticulously configured as a complex lipid matrix integrating oleic, palmitic, palmitoleic, and stearic acids to emulate the multi-component nature of recycled cooking oil. Residue 2 was restricted to stearic acid to serve as a high-pressure benchmark, putting a metabolic stress scenario on the table.
With these scenarios established, we perform a flux balance analysis to visualize the flow of metabolites. For residue 1, we decided to elevate the total flux to negative 20 millimoles per grams of dry weight per hour, since our goal here was to evaluate the maximum metabolic capacity of the model by simulating saturation and metabolic stress. By doubling the molar flux compared to our glucose control, we forced the model to manage an excess of carbon atoms, thereby stressing the cell's redox balance during beta oxidation. And this environment is crucial because it forces the network to find alternative routes to generate NADPH, which is essentially the power needed to synthesize PHA. And for residue 2, which consists of pure stearic acid, we used a flux rate of negative 10 millimoles per gram of dry weight per hour, and this provided a control baseline to compare it directly against our glucose medium, which is the control, and the results were really clear. Lipids are a far superior feedstock, as shown in our product comparison table, which is here. While glucose might only produce about 2 units of acetyl-CoA per unit of uptake, the lipid-based process can generate significantly more, giving us a massive stoichiometric advantage against the common carbon source, which is the glucose. Continuing with our model, we made an analysis on how each substrate impacts both on acetyl-CoA and NADPH production, given 10 units of each oil, in order to verify the efficiency of each fatty acid found in the residues. In both analyses, the same three fatty acids show their potential as favorable substrates for these molecules, being the top three oleic acid, stearic acid, and palmitoleic acid, and they showed a better yield than glucose. To conclude our project, the mathematical modeling allowed us to prove that we can take common pollutants such as waste cooking oils and turn them into high value resources. Through our flux balance analysis, we found that lipids from household and food industry waste are far superior to traditional glucose, being oleic acid the one outperforming glucose in both acetyl CoA and NADPH production, two essential ingredients for bioplastic. Our model shows that the best design isn't just expressing genes harder, By using orthogonal control, which is balancing the precursor supply against the polymerization process, we can favor the production of fewer but longer high quality polymer chains. We also took a look at the metabolic burden. Our analysis warned us that aggressive gene expression can cause the system to collapse, with relative stress indices spiking over 90%. However, by tuning the system to a workable stress level of about 50 to 60%, we found a stable sweet spot. This mutant strategy is the key to scalability. It predicts a 128 grams per liter yield and a nearly fivefold increase in storage capacity. Also, under the guidance of Dr. Michelle Abraham Ruiz Robles, our research is heading in three clear directions. First, using molecular dynamics to refine the physical chemical parameters of our polymer. Second, formulating PHB copolymers to optimize their mechanical properties. And finally, running electrospinning simulations for clinical validation.